Hello everyone, today we're going to be making elephant toothpaste, and these are just the materials on the screen as you can see. So we're just going to go over what we need. Some hydrogen peroxide, around 3% or 6%, some cups to pour the solution in, and main ingredient is active dry yeast. Make sure it is dry yeast. Some food coloring, some soap, dish soap would be preferred. A beaker, it could be bigger than this, it doesn't matter. A spoon to mix anything, and the hot water. So, and you can't forget to have a tray. That part is optional. So what you want to go ahead and do is take your dry yeast and you want to pour it in a cup. You want to add around like a tablespoon or 2 tablespoons. It just depends on how big the beaker or cylinder is that you're gonna be pouring in. So from now we're gonna go ahead and add hot water. I'm adding the same amount of hot water that I added of dry yeast, so I'm putting around 2 tablespoons. After you put the water in and the dry yeast, you just want to go ahead and mix it up, and you just want to stir it for about like a minute, 30 seconds to 1 minute, until you start getting like a thick consistency. So after mixing it, you can see it's starting to get thick. You just want it around this type of consistency, just somewhere around that. And now we're just gonna go ahead and take our aluminum foil cover, and just place it down so there's just less mess for us to clean up. And you want to go ahead and take a glass beaker or a cylinder, or it could be even a bottle, it's up to you. Just place it. So the first ingredient we're going to add in here is hydrogen peroxide. I am using 3%, so like you will have to add just a little bit more because it's it's not as strong as 6% per se. So if you do have 6%, that will work, and you'll just get more of a reaction, which will be even better. So I added around half of the beaker of 3% hydrogen peroxide, and now I'm adding a little bit of food coloring. You can add the powder type like the one I'm using, or you can add like droplets. It's up to you. This is just what I had available. You want to go ahead and stir the food coloring in, just so we have like a constant color of when the foam is appearing to look at. And after adding in your food coloring, you want to go ahead and take some dish soap. This here is just regular dish soap I had. You can use any type, doesn't matter, because you just need a little bit, like a few droplets, a few seconds of it, and you just want to go ahead and add it in there and stir a little bit. After stirring it, you want to go ahead now and take your solution from earlier of the dry yeast and hot water and go ahead and pour it into the hydrogen peroxide. And from here you should go ahead and see the foam appearing out of the beaker. As you can see, the aluminum foil plate is just starting to be filled with foam. Now, if you are using a stronger hydrogen peroxide, if you have that available, the foam will just come even at more speed and more of a rate. So that would give you better of a result. But I just happen to have 3%, and I just poured in about half of it. So you can go ahead and see that the foam is just slowly still continuing to come out. And now we're going to talk about why this happens, the science behind it. Alright, so now we're going to be looking at the science part on how this actually works. And I'm going to try my best to break it down on what happened in the experiment. So how exactly does having hydrogen peroxide, soap, food coloring, and then yeast and warm water equal the foam? So the main ingredient, or the main reason this happens, or one of the major parts, is yeast. So when we talk about yeast, yeast we know is a living organism. They're single-celled, small microorganisms, and they reproduce and everything. So yeast are actually living organisms, and yeast also in this experiment, or almost in any other experiment, are something that we called catalysts. Now catalysts are some chemicals, or another product that increases the rate of a chemical reaction. So in this experiment, dry yeast was the living organism. It was a catalyst that helped increase the rate of a reaction, which led to the foam coming out really fast. So we're gonna break down how this happens in the next slide. So just know that a catalyst is a substance that increases the rate of a chemical reaction. So in this reaction, the yeast, which are living organisms, mixed with the hydrogen peroxide, and we're going to see how it breaks down in this slide right here. So the enzyme catalyst breaks down the hydrogen peroxide, as you can see here. So the enzyme catalyst was the yeast, and the yeast breaks down the hydrogen peroxide into H2O and O2. So we start here with H2O2, which is the chemical formula for hydrogen peroxide. So when the hydrogen peroxide comes in contact with the yeast breaks it down into H2O and O2, which is oxygen gas and water. So that's why you can see in the experiments that when the reaction happened, the top was just all foam, and that was just the hydrogen gas coming out. And the inside of it, if you were to go ahead and put your hand in it, it wasn't hydrogen peroxide anymore, it was straight water. So the hydrogen peroxide was broken down because of the catalyst here, and that increased the reaction, and it happened really, it broke it down really fast, and it split into H2O, which is water, and oxygen gas. So the foam on top wasn't hydrogen peroxide, it was straight oxygen gas trapped in the foam bubbles. So when we look down at it, it's a very simple experiment, and it shows the rate of reaction here, what yeast plays, and it shows it very well. This is a safe experiment for anyone to do, and it was definitely a fun one for me to do. So just going over again that the yeast contains an enzyme called catalyst, and that's what broke down the hydrogen peroxide into oxygen gas and water. So the oxygen gas gets trapped in the by the soap. So the soap, what the soap does is traps the oxygen gas. So the soap mixes with the oxygen gas, which is the large foamy solution that's on top. And catalyst can be found in almost anything, and one of the main things it's found in is potatoes. We learned that in biology. So overall, yeah, this was a fun experiment to do, and here it is broken down. So there you go.